Hello students, let's study antigen antibody reaction. Antigen antibody reactions in vitro are known as serological reactions. They help in diagnosis of infections, epidemiological surveys, identification of infectious and non-infectious antigens. Measurement of antigen and antibody titer or units. So antibody titer of a serum is the reciprocal of the highest dilution of serum, which shows an observable reaction with the antigen in the particular test. The important parameters are sensitivity and specificity. Sensitivity, ability of the test to detect even very minute quantities of antigen or antibody. So false negative results are absent or minimal. Specificity, ability of the test to detect reactions between antigen and its homologous antibody. So false positive reactions are absent or minimal. Murach's or lattice hypothesis. So it is zone phenomenon. Antigen antibody reaction is weak or fail to occur when the number of antigen and antibodies are not proportionate to each other. So first is prozone phenomenon. In the earlier test tubes, antibodies are excess. Hence, antigen antibody reaction does not occur. So it is antibody excess. Post-zone phenomenon in the later test tubes, antigen is excess. Hence, the antigen antibody reaction failed to occur. So post-zone is the antigen excess. So in prozone or post-zone, lattice does not enlarge due to inhibition of lattice formation by excess antibody or antigen respectively, as the valencies of the antibody and antigen respectively are fully satisfied. So in the zone of equivalence, there is the optimal proportion of antigen and antibody. So there is the lattice formation. Types of antigen antibody reactions. So conventional and near techniques. So in conventional techniques, it is precipitation, agglutination, complement fixation, and neutralization. In newer techniques, it is enzyme link immunosorbitase, that is ELISA, enzyme link fluorescent assay, then immunofluorescence assay, radioimmunoassay, chemiluminescence linked immunoassay, that is CLIA, immunohistochemistry, rapid test, lateral flow, immunochromatographic test, flow through, then Western blot and immunoassays using the electron microscope. Precipitation reaction when a soluble antigen combines with its antibody in the presence of electrolyte sodium chloride at a suitable temperature and pH. The antigen antibody complex forms an insoluble precipitate is called precipitation reaction. So, and then precipitation in liquid medium forms the insoluble floccule is called flocculation. The application of this precipitation reaction, VDRL test for diagnosis of syphilis, RPR test for diagnosis of syphilis, Elecgel precipitation test for diphtheria toxin. Second, agglutination reaction when particulate antigen is mixed with its antibody in presence of electrolytes at a suitable temperature and pH. The particles are clumped. The agglutination is more sensitive than precipitation for detection of antibodies. Mechanism when antibody is added to suspension, it combines with surface antigens and links them together to form clearly visible aggregates. Types direct agglutination, so it is antigen detection, and in that it can be done by slide and tube method. So a slide example Vidal test and tube example Vidal test for determining the titer. Indirect agglutination, detection of antibody, so passive uh, antibody detection, latex agglutination, example ASO, reverse passive antigen detection, latex agglutination method, example RACR. Hemoglobination, so RBC is used as an antigen. Example, pulmonary test for extreme war virus detection, blood group use test. Third is a complement fixation test, and fourth is a neutralization test. So in that, toxin antitoxin neutralization is there. Example is the Nagler reaction used for detection of alpha toxin of the Clostridium perfringens. Precipitation reaction, we have already seen the definition. Precipitation in liquid medium, ring test in a narrow tube, example capillary tube, antigen solution is layered over the antiserum. Precipitate ring appears at the junction of two liquids, example streptococcal grouping by Lancefield technique. A SCOL is thermoprecipitation test for anthrax. Flocculation test when a drop of antigen is mixed with a drop of patient's serum, floccules appear, example VDRL RPR. Tube flocculation can't test previously used for the syphilis. Precipitation in gel is also called immunodiffusion. Using 1% soft agarose gel for precipitation reaction, many advantages over the liquid medium. Results in formation of clearly visible bands instead of flocules that can be preserved for longer time. Can differentiate individual antigen from a mixture as each antigen forms a separate band. Principle whether only antigen diffuses, single diffusion, or both antigen antibody diffuse, double diffusion. Whether antigen or antibody diffuses in one dimension, that is vertical diffusion when test is done on tube layer with gel. Two-dimension diffusion in both x and y axis when test is done on slide or petri dish layered with the gel. Types of immunodiffusion in gel. So first is single diffusion in one dimension, Odin procedure. Double diffusion in one dimension or clay through procedure. Single diffusion in two dimension, radial immunodiffusion. Double diffusion in two dimension, Orther-Laning procedure. Example of two, this is Legges detecting diotoxin of Corynebacterium diphtheria. Agglutination reaction when particulate or insoluble antigen is mixed and then it forms the clumps. So in the types, first is direct agglutination. So in this, antigen directly agglutinates with the antibody method, slide and tube method, and then uses a typhoid fever, Vidal test, acute brucellosis, standard agglutination test, Coombs agglutination antiglobulin test, heterophile agglutination test, example, typhus fever, that is Will-Felix reaction, infectious mononucleosis, Paul-Bunel test, mycoplasma pneumonia, cold agglutination test. Indirect or passive agglutination test for antibody detection. Then examples are indirect hemagglutination test, latex agglutination test, and passive agglutination test where RBCs are used as a carrier molecule. Let's study serological tests like RAC-RP, SOVID, RBD,
Hello students, today we will see serological test. So first is RA test. So it is rheumatoid arthritis test. So clinical significance is RA test is done to detect presence of rheumatoid factor in patient of rheumatoid arthritis. So rheumatoid arthritis is a disease of the old age patients in that all the joints are major joints are involved. Procedure, in short, you have to take three circles, label it as test, positive control, negative control, add serum to test, then positive control 1 drop to PC and negative control 1 drop to MC, and then add antigen provided in the kit, mix and wait for 2 minutes and then see for the clumps. C-reactive protein CRP, it is also done same way. Take taste, positive control, negative control, and then add serum and then reagent and mix and wait for 2 minutes and see for the clumps. So C-reactive protein significance, it is mainly a inflammatory marker of inflammatory condition of infectious or non-infectious origin. So when the WC count is raised, ESR is raised, CRP is also positive. Antistreptolysin O titer, that is ASO test, it is done for the investigation of rheumatic fever. It is a post streptococcal disease. In that, antistreptolysin O titer is raised and procedure is same as RA and CRP. You do take three circles, taste positive control, negative control, add serum, PCNC and add reagent and then mix and wait for 2 minutes and see for clumps. Thank you. Hello students, today we will see serological test, that is Vidal test and VDRL test. So Vidal test is done for diagnosis of the typhoid fever and paratyphoid fever, that is caused by Salmonella typhi and paratyphi A and B. So in that, what is the principle? So in that, four antigens are there: O, H, AH and BH. So O is pink color, sorry, O is blue color and H, AH, BH are pink color reagents. So procedure by slide method, you have to add 1 drop of serum, 1 drop of positive control, 1 drop of negative control, and then you have to add, so the total 4 drops you have to take to add 4 antigens of the serum, and then add 1 drop of O, H, AH and BH antigen, mix and then wait for 1 minute and then see for the clumps. So if clumps come within 1 minute, then it is the positive test. So O and H for the typhoid fever, AH for the paratyphoid A fever, and BH if clumps are there, then it is for paratyphoid B fever. Then VDRL test, so it is venereal disease research laboratory test, method by slide method. Again you have to take 1 drop of serum, 1 drop of positive control, 1 drop of negative control, then add antigen to each one drop, and then mix and rotate for 8 minutes on the VDRL rotator or by manually, and then see for the clumps. So here you will see floccules. So report can be done if no floccule seen, non-reactive; if weak floccule seen, weakly reactive; if strong floccule seen, it is strongly positive. And then significance, it is done for the diagnosis of the syphilis test, and syphilis is caused by Trypanosoma pallidum spirochetes. It is a sexually transmitted disease, and then it should be done usually in pregnancy to rule out congenital syphilis, because in that baby may get stillbirth, IUD, or mental retardation. So that syphilis test is done. Thank you.
Hemoagglutination test, where the RBCs are used as a source of antigen in agglutination test. Then application pulmonary test, cold agglutinin test, blood glucose Coombs test for antiglobulin test. Coombs test or antiglobulin test is performed to diagnose Rh incompatibility by detecting Rh antibody from mother and baby serum. So in Rh incompatibility, Rh negative mother with Rh positive baby because of the Rh positive father, and then if the Rh antibodies are incomplete or blocking antibodies. So there are two types, direct and indirect. So in direct antiglobulin test, the Rh antigen is present on the RBCs, and the other RBCs are antibody coated on fetal RBC. So antiglobulin added, that is Coombs reagent, and then Rh antibody coated fetal RBCs get agglutinated. Indirect antiglobulin test, there are free antibodies, Rh antibodies in mother serum, and then reagent RBCs are there. When Coombs reagent added, the Rh antibody coated on reagent RBC gets agglutinated. Complement fixation test. So as the name detects antibodies in patient serum that are capable of fixing with the complements. It was very popular, but now it is obsolete. Application Wassermann test was also widely used for detection of complement fixing antibodies in rickettsia, chlamydia, brucella, mycoplasma infection, and some viral infections such as arbovirus, rabies. Complements are also used for various serological tests other than CFT, like Treponema pallidum immobilization test, Sabin Feldman dye test for toxoplasma, and Vibrio antibody test. Neutralization tests nowadays most not commonly used. Various examples: virus neutralization test, plaque inhibition test, toxin antitoxin neutralization test. Examples: Schick test, Nagler reaction, ASO test, hemagglutination inhibition test. Antibodies in patient sera can agglutinate with the hemagglutinin antigens present on surface of some viruses. Was used in past for diagnosis of various viral diseases. Example: influenza. Newer techniques. The first one is the ELISA, enzyme link immunosorbent assay. So principal antigen antibody complex attached with the enzyme plus substrate activates chromosome, and then color changes there, and that is detected by ELISA reader. Enzyme is the peroxidase, immunosorbent is a polystyrene or polyvinyl material, substrate is the hydrogen peroxide. Types of ELISA: direct ELISA to detect antigen, indirect ELISA to detect antibody, sandwich ELISA to detect antigen, competitive ELISA to detect antigen. Application: sensitive test to detect antigen antibody reaction for HIV, hepatitis A antigen, hepatitis B, E antigen, HCV, NS1, dengue antigen, Epstein-Barr virus antigen, HSV, toxoplasma. Advantages: test is specific, sensitive, large number of samples tested at one. Mostly used in the blood for the screening of the donors. Disadvantages: costly. Enzyme link fluorescence assay, modification of ELISA. So it is automated system, and antigen antibody complex is detected by fluorometric system. Application: markers of hepatitis viruses, HIV, antibody to torch, measles, mumps, rotavirus, and H. pylori. Immunofluorescence assay. Fluorescence refers to absorbing high energy short wavelength ultraviolet light rays by the fluorescent compounds and in turn emitting visible light rays with low energy, longer wavelength. Fluorescent dye is used to conjugate the antibody, and such labeled antibody can be used to detect the antigen or antigen antibody complex on the cell surface. Types: direct, indirect flow cytometry. Flow cytometry is laser-based technology that quantitatively analyzes and separates the cells as they pass through the laser beam. Application: detection of autoantibodies, antinuclear antibody in autoimmune diseases, detecting microbial antigen, the example rabies antigen in corneal smear, then detection of viral antigen in cell line inoculated with the specimen, flow cytometry, CD4 count in the HIV infected patient, detection of leukocyte with specific marker for diagnosis of various lymphomas. Chemiluminescence linked immunoassay, that is clear. Emission of light as a result of chemical reaction. Light is detected by luminometer. The application: detection of antigen, antibodies, hepatitis viruses, HIV, and torture. Immunohistochemistry to detect antigen in cells of tissue section by using labeled antibody. Diagnosis of tumor cells. Western blot detects specific proteins or antibodies in a sample containing mixture of antibodies which targeted against different antigens of same microbe. Procedure: so first SDS page, separation of antigen fragments, then nitrocellulose membrane plotting, and third is the enzyme immunoassay. So used as a supplementary test to confirm HIV, Lyme disease, HSV, and toxoplasma. Rapid test immunochromatography, lateral flow assay, colloidal gold is used for labeling antibody. Application: hepatitis surface antigen, dengue test, rapid malaria antigen test. Flow through assay, protein A is used as labeling antibody. Example: Tridot, HIV, and HCV. Montux test, in vitro detection of sensitization by Mycobacterium tuberculosis. Montux test is performed by intradermally injecting 0.1 ml of tuberculin PPD of tuberculin by tuberculin syringe. For single dose test, 5 TU is used. Uses the diagnosis of suspected infection by Mycobacterium tuberculosis in young children as a post-vaccine check for the efficacy of BCG vaccination. Enzyme link immunosorbent assay. So there are two types, homogeneous and heterogeneous. So in homogeneous, all regions are added at one step and then used for detection of haptens like drugs. And then heterogeneous, so named because of two components, immunosorbent and enzyme. And there is substrate enzyme, the chromosomal system added to the final step of ELISA. ELISA principle, antigen antibody complex, enzyme, then substrate is added which activates the chromosomes, and there is color change detected by spectrophotometry. And here you can see ELISA reader, ELISA washer, and automated ELISA machine by BioRad. ELISA is performed on microtetraplate containing 96 wells, micro ELISA, and sometimes in tubes, that is macro ELISA. So microtetraplate or tubes are made up of polystyrene or polyvinyl by polycarbonate material. ELISA kits are commercially available, and then procedure involves a series of steps done sequentially, and each step reagent is being added, incubated, and followed by the washing. Types of ELISA: direct, indirect, sandwich, competitive, and ELISpot. Direct ELISA used for detection of antigen in test serum. Primary antibody targeted against serum antigen is labeled with the enzyme. Step 1, well of microtetraplate are empty, not pre-coated with the antigen or antibody. Step 2, test serum containing antigen is added into the wells. Antigen becomes attached to the solid phase by passive adsorption. Step 3, after washing, the enzyme labeled primary antibody raised in rabbits are added. Step 4, after washing, substrate chromosome system is added and the color is measured. Indirect ELISA, antibody detection. So solid phase of wells of the microtetraplates are pre-coated with antigen. Wells are coated with antigen plus primary antibody, that is test serum, plus secondary antibody labeled with enzyme plus substrate chromosome development of color. Indirect ELISA antigen detection wells are empty, not pre-coated. Antigen, that is says serum added, gets adsorbed to well plus primary antibody plus secondary antibody enzyme labeled. Then substrate chromosome development of color. Advantages of ELISA. So it is method of choice for detection of antigen antibody. Sensitive, specific. Disadvantage. It is costly. Application. ELISA used for antigen detection. Hepatitis B surface antigen, pre-core antigen, that is HBe antigen, NS1 antigen for dengue. ELISA can also be used for antibody detection like hepatitis B, hepatitis C, HIV, dengue, EBV, HSV, toxoplasma, leishmania. Enzyme link fluorescence assay. So it is modification of ELISA because it is fully automated system. All steps are performed by instrument. Antigen antibody complex. Antigen antibody enzyme complex is detected by fluorometric method. VIDAS and mini VIDAS are the systems available. Advantage automated system to perform easy, user friendly, less contamination, give quantitative results, more sensitive and specific. Disadvantage expensive. Application infectious diseases, biomarkers like procalcitonin, hormones, tumor markers, cardiac markers, screening of allergy. Immunofluorescence assay, similar to ELISA. The difference is fluorescent dye is used for the labeling of antibody and detects cell surface antigen or antibodies bound to cell surface antigen. Then it is absorbing high energy short wavelength ultraviolet light rays by fluorescent compounds and in turn emitting visible light rays of low energy longer wavelength. And then types are direct indirect application detection of autoantibodies, rabies antigen and viral antigens on cell lines. Flow cytometry, laser-based technology that quantitatively analyzes and separates the cells as they pass through the laser beam. And then application is CD40 cell count in HIV infected and leukocyte antigen or markers on the lymphoma. Western blot, it is the detects specific proteins in sample containing mixture of antibodies. Southern blot detects DNA fragments, northern blot detects mRNA, eastern blot detects carbohydrate epitopes on proteins or lipids. Application supplemental test for the HIV. Rapid test, a point of care test. There are two types, lateral flow through. Immunochromatography, that is lateral flow assay. So the test system consists of nitrocellulose membrane and absorbent pad. Two formats are available, cassette and strip. Application hepatitis B surface antigen. Second method in ICT is the flow through assay. So protein A is used for labeling antibody. In lateral flow, it is gold conjugate. So sample flows vertically through the nitrocellulose membrane, and then example can be antibodies to HIV. This chart is a summary of the newer techniques. So ELISA, in that color change is detected by spectrophotometer, then enzyme link fluorescence assay, fluorometry detection, immunofluorescence assay, emits light and detected by fluorescence microscope, radioimmunoassay, emits beta and gamma radiation and detected by counter, chemiluminescence linked immunoassay, emits light detected by luminometer, immunohistochemistry, color change, naked eye or fluorescence microscope, Western blot, color band, immunochromatography, that is from lateral flow and flow through color band measured by naked eye.
Hope you find this session useful for your understanding and for your exams. Thank you.